Okay, good afternoon. Before we begin, actually, I'm a little bit concerned that probably by now a lot of you must been wondering what's going on the grading of the midterm exam. So I've been extremely tied up lately, so I wasn't even able to start grading yet, to be honest. So please, if you please provide me a little more patience after I get all these my things over quickly, and I will just jump right on to the grading as soon as possible. So that's what I can tell you now. And also, for those who are the international students, that at least a couple of international students are going to take for the final, take the take-home exam, the mode of the exam, because they are not going to be graded equally as your regular students. So it doesn't matter because of their schedule conflict of the flight back home schedule. So what I'm going to say to all those international students is, if any other additional international student also wish to take the take-home exam instead of in-class exam for the finals, let me know, then I can arrange those for you. The take-home exam will be like a written form of, I will give you 1 week, okay, and then you can write the answer and then submit through email. That will be the way format. So if you rather take that route, then let me know, okay? All right, so today is all about Watson and Crick and their result, which revealed the DNA double helical structure correctly. It was the one actually finally put an end to all this ongoing debate over which was the genetic material, protein or DNA on chromosome. Interesting me, actually. With all those experiments, marvelous, meticulous experiments that I have introduced briefly in the previous lectures, the people were very skeptical, stubborn and skeptical to accept the result. But all of a sudden, this result, even this one, was eventually, because of this work, Watson and Crick received the Nobel Prize several years later in 1962, like almost 10 years later. But surprisingly, this was so famous. This result was one exception that any scientist who won in the life science, who won the Nobel Prize without performing a single experiment. They didn't do any single actual experiment. Simply it was a brain work, conceptual work. So there was really irony of. Anyway, so these 3 sentences are all self-explanatory about what they have done about this double helical model. But then, actually more importantly, there are 2 predecessors that somehow worked, at least to some proportion, worked on this DNA, somehow knowingly or even unknowingly involved in this business. These are, one was the information as a result of their work. Still, once again, why this one is not following? We have already. Okay, now, okay. This is a Chargaff's. One is called the Chargaff's rule, and the other one is this X-ray diffraction data. That was also another very famous experimental data made by one young woman scientist named Rosalind Franklin at the time in Britain. They were actually, Watson and Crick and Rosalind Franklin's lab was like right next to each other. So they were like friends and also competitors. towards the elucidation of the structure of the DNA double helix. Anyway, these two result heavily been able to influence the outcome, the result of the final Wasserman Crick. So first, let us take a look at the what was the Shagap's rule. Irving Shagap was actually a very well-respected biochemist at the time, and been working in the United States, and he apparently, as we look at his remark over here, his general attitude over the molecular biologist was like this, scoffing at, like despised the work of the molecular biologist, because to them, to the biochemist, molecular biology was something new branch of the science, and then it seems like a lot of portion of this, or the area where biochemists work and the molecular biologists work overlapped actually. So when actually, if you if you're going to take the biochemist course in your near future, you will soon realize that a lot of those material, the content in the textbook as well, is also overlap of those of molecular biology. Usually, we can say that molecular biology somehow branched out because biochemistry has a longer tradition than molecular biology. So this molecular biology is a little bit newer thing. So to him, probably this remark must have been out after the result of this Wasserman Crick's the DNA double helix structure, and then he realized that it was actually his inspiration that made those things. So maybe a lot of them, partially at least, was out of jealous of this harsh remark about this. Molecular biology is essentially the practice of biochemistry without a license. They are just nobody, and we are the original. Is kind of at least his sentiment at the time, maybe. So anyway, so what is about Shagap's rule? Again, I cannot really. Is that he examined all different living organisms and the relative ratio of DNA nucleotide. Remember, DNA is a polymer made of what? What's the individual components? What do you call the individual chemical components that make DNA? Anybody still remember? The individual chemical component being assembled to finally make DNA strand. That individual component, we call them what? Start N? Yes, thank you. Nucleotide. Nucleotide is the chemical component. There are only 4 types of nucleotide. So DNAs are like all just jumbled mixture of this in seemingly random order of A, T, C, G. When we call those A or T or C or G, that is simply the type of particular nucleotide, abbreviated form of particular one of the 4 different nucleotides. Okay, so he compared the ratio, all those ratio of nucleotides, because there are only 4 different kinds of nucleotides present in whatever the organism you examine. Okay. And then he found some very strange, interesting the ratio. So no matter what, obviously their overall nucleotide ratio was something like, will be different, unique, depending on different organisms. However, no matter how they are different, always this ratio, like the ratio between purines and the pyrimidines. What? What's purine and what's pyrimidines? More specifically, the amount of T always equals to the amount of the A nucleotide, and the amount of G nucleotide is always equals to C. And what I'm saying is purine and pyrimidines are basically this. DNAs. This is very simplified, very simplified cartoon of one single nucleotide over here. This is one single nucleotide. Okay, so actually this single nucleotide consists of 3 individual parts. Here is one part is a sugar, carbohydrate. The other part, P, standing for P, is a phosphate, a derivative of a phosphoric acid, a phosphate. Okay. And the third part here we call is a nitrogenous base because it acts like some alkaline base, chemically speaking. So all these 4 different nucleotides are shown over here. 1, 2, 3, 4 different types of possible nucleotides. Simplified structures are shown in this cartoon, in that among those 4 different types of nucleotide, the only different portion is where the nitrogenous base area. That's the only different part. The other parts are all identical for these 4 different types of a nucleotide. So this particular type, so 1, 2, 3, 4 different types of this chemical portion, we call the nitrogenous base. This actually is a part of the chapter 3 that as part of your Bogang. Obviously, nobody didn't pay any attention of listening or viewing that part. That's why the facial expression, what is, okay. Actually, this should have been already something that is familiar to all of you if you followed that, if you took that Bogang course at home. But anyway, so these are 4 different nitrogenous bases which are part of all this nucleotide. And if you look at it, the reason why they are different, there are fine chemical differences which are not revealed in this very simplified version. But anyway, it's called the guanine, this type. The one that's being colored in purple over here, this nitrogenous base is called the guanine. Okay, that's why this particular nucleotide can be abbreviated as a G usually, because other than this part, the rest of the thing is simply identical to the others. So you can simply recognize this by its unique part, which is a guanine. So guanine starts with G, so we usually casually call this, this is G. Okay. When we deal with the nucleic acid sequence, the G, rather than guanine, then it's much more convenient and efficient. So this G. And same token, C stands for cytosine. There is another nucleotide called cytosine. And T, thymine. A, adenine. But if you look carefully, you notice that these 4 different types of nucleotide of this nitrogenous base, there are 2 different kinds. Can you recognize? 2. One is what? Having 2 rings. Among this, according to speaking of this nitrogenous base, there are 2 kinds. One nitrogenous base kind consists of 2 these lines, all these lines in chemical structure represent the covalent bond or chemical bond. And all this, each point, carbons are missing. So this is what basically simply carbon, organic compound, also having some of the nitrogens. Basically, its frame is made of all carbon, but one nitrogen, one nitrogen, one nitrogen, one nitrogen. That's why it's called a nitrogenous base, right? So anyway, one such nitrogenous base can have this 2 carbon ring, whereas another group of nitrogenous base we call as a pyrimidine consists of only 1 ring, such ring. So when we call pyrimidine and purines, that's exactly what we are talking about. So there are, what 2 nucleotides in DNA are purines? Yeah. Guanine, yeah, A and G, these are purines. Good. And the T, thymine and cyrosines are pyrimidines. That's it. And what this Shagab has discovered was simply this. Simply all this ratio, all those all the purines in DNA of one organism means all those A plus G, right? All together. And all those pyrimidines, T and C's, their ratios are always 1 to 1, obviously. Yeah. Why? Because they have their special rule of pairing. Always the A nucleotide will pair with T only, and G with C only. They have their own agreement. This G all of a sudden does not pair with A. It's no. This G does not even pair with T. The G always have its own partner with C only, and everybody else in the nucleotide will do the same. That's we call complementary base pairing. That's also appeared and also emphasized in your previous lecture, Bogang. Okay, but anyway, so these are the, so what he have actually discovered without even realizing, was this golden universal rule of DNA. DNA exists in double helix. And the reason why DNA exists in double strand is because the individual nucleotides that make DNA have this specific pairing, because the chemical bond, H bond, what are H bond? It's not H bomb. Someone mentioned today a bomb, some kind of bomb. So, that is the reason why you mentioned the bomb, some kind of bomb today. But this is not a bomb, bond. So just like the weak bond, it's not covalent bond, but hydrogen bond. This is a hydrogen bond, which is very weak, easily breakable, and also, although it is weak, easily breakable, but also at the same time, it's easily can be formed again. That's the beauty, advantage of this type of a weak chemical bond in life. So because of that, yeah, you can easily understand that in every different organism, if you compare this different ratio of nucleotide, it always matches. Like all those purines plus all those pyrimidines, because this is the rule. And in particular, more specifically, this type of rule, like always T and G, has the same number, because these are the 2 couples that establish and maintain the partnership of nucleotide. Okay? So that's what he had discovered, the Shadow's rule. But he was not able to explain why this rule was found in every different organism. Simply, I don't know why, but it's very funny, isn't it? But no matter which different kind of organism you look at it, you will find exactly the same rule over here. So that's why people, oh, yeah, that's right, you're right. So you don't know the reason? No, I don't know. Okay, fine. But that's always like it applies, so we will call it the Shagap's rule. That's what it is. So this is one actual data of the Shagap's rule. The one that is very small, so it's difficult to read, is the information, including that actual DNA, different nucleotide proportions are all uniquely different in different organisms. Like when you compare different organisms like E. coli bacteria, like inside of your gut, and Mycobacterium is another bacteria, yeast, Drosophila, you know that Drosophila, and corn, and calf, pig, human, all, wherever you look at it, this whole ratio or approximate. This is actual data, so it may not exactly equal to 1 exactly, but it's almost good as the equal 1, purine versus pyrimidine. And also AT-GC ratio also reaches all 1, right? There's only exception, if you have a very keen eye, this. Exactly, good question. Not all bacteriophage have this kind of deviation. All other bacteriophage will also follow. But this 5X174 bacteriophage is something very unique in that this is SS DNA. What do you mean SS DNA? Don't think about that. It's not that SS, okay? SS is a single strand. So DS, oh shoot, please, don't do that. DS is double-stranded. So some bacteria exist as just a single. It's funny, strange, but yeah, that's right. Some bacteria exist as a single strand genetic material, and then they enter. After entering the host, and they temporarily restore this double-strandedness to do all this different job that need. So in this case, single means each nucleotide doesn't have any partner. So there is no room for this Shagap's rule could apply. That's why you see this deviation from this. Okay, but so this has to be an exception. Hey, what's going on? Oh, I broke. That's why. Okay, it didn't break. All right, so what happened was this. The complementary base pairing between the nitrogen bases was the Shagap's rules, the reason for the Shagap's the rule. But he himself did not know. Simply, I don't know reason, but blah, 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 there's a rule, so just honor me. Okay, fine, credit goes to you, but we still don't know what the reason. But these wise men kind of got a clue about, probably that means something. Okay, so probably this is something, this particular, because they also knew DNA is made of these 4 different types of nucleotides, and they also knew that if you examine the individual nucleotide chemically, then there are some rooms that specific interactions, a specific chemical bond, like maybe hydrogen bond, they all know about. Chemistry, the field of chemistry was far ahead of the molecular biology back then. So chemically speaking, they all already analyzed and knew specifics at the time. So it's simply how do you put those different chemical information together to explain the lives, the different interesting phenomena. That's what they did, Watson and Crick. Okay? So they thought maybe this Sharap's rule was because this particular interaction, specific interaction between specific nucleotide, was the one responsible for these two strands holding together. Okay? And another support as a clue also came from, by the way, before we say goodbye to Irving Sharap, although he mentioned such a something a little bit stupid sound comment about molecular biologists, but if you read this, okay, that is something fine. I can just give my respect back to him. He also said science is wonderfully equipped to answer the question how. Yeah, I agree. But it gets terribly confused when you ask the question why, actually, impatiently. Many occasions, actually, when you are not ready in science, simply try to ask question why, then you're not going to get anywhere. But you begin to start questioning how first, stepwise, and then your why, your question of why become more elegant at the time. So anyway, so now the second clue came from this famous Rosalind Franklin X-ray diffraction data of DNA. This is what is X-ray diffraction? This technique, although developed very early in early 1900, so during the 1950s, people were able to use this technology, but this more sophisticated, developed, advanced version of this X-ray, we call the X-ray crystallography in these days, was the one actually being utilized to decipher the protein structure, molecular structure, which is a very important tool even in these modern days of molecular biology too. But anyway, the principle, briefly explaining this principle is that, let's say you have this sample, whether DNA or whatever the sample, fine. It could be protein or any big object. Then you shoot the beam of X-ray at high energy, and then it collides with this object and then has some kind of scatter, some diffraction pattern. That's why it's called the diffraction. And in the X-ray film, all this X-ray will have its own unique footprints. And if you are an expert, by this pattern, you can kind of deduct the actual shape of this very small molecule like DNA. So, if this X-ray photograph was presented to you, what is this? You cannot even interpret it. But then, oh, this is a helix type of, yeah. This is some kind of helix, and the pattern of a helix, the repetitiveness, like some bases will appear every, this helical turn will have about 10 bases type of thing can also be obtained from this type of. In more advanced class, I could even go along with actual interpretation of the X-ray crystal or X-ray diffraction data, but here, no. It's out of our actual scope. So she, with her graduate student, these two team together reported two actual famous the photograph of DNA molecule using X-ray diffraction. And this among them, this one was more famous. And this one was the one actually Watson and Crick get the inspiration from. But also because depending on some of the fine chemical condition, physiological condition, DNA structure can be different. But even Rosalind have resolved these two alternative form of DNA structure. Nowadays, we recognize the A form DNA and B form DNA, and these can exist interchangeably in our cell too. But for the type of standard shape that Watson and Crick presented was actually out of this B form DNA data. Anyway, so the summary of the Watson and Crick's DNA structure was this. The one is DNA exists in double strand because the importance is this. Out of this Rosalind Franklin's the X-ray diffraction data does not directly indicate that, oh, the DNA is a double helical structure. It is a helical structure of either double strand or it could be triple helical structure. You don't know. So you have to figure it out through another several lines of more thorough experiments. That's what this Rosalind Franklin did it. So she, after getting this result, Rosalind thought, oh, it could be double or triple helix, so I will have to sort this thing out by further experiment. I will do this. So overnight, she has done this. But Watson and Crick that actually stole her data. They didn't have actually official permission of getting access to her data, but simply her boss, his name was Maurice Wilkins, and the Watson and Crick, hey, can you store her data so that we can just analyze, so that we can just go this direction? And he did, oh, sure, I can. And so unofficially, I have an access to this data, and then they also, it could be figured out that it could be double or triple helix. But what I have taken differently is instead of doing this experiment, but they, we can afford doing this, but figuring this thing out through experiment. So let's take a guess. And they have another clue, which was the Shagap's rule. So I think they said, they were like, we think this could be, it could be, of course, it's a triple helix too, but it could be more like a double helix. Well, let's take a guess, and based upon this double helix, let's build a model. Sometimes simple such wise guessing expedite your route. So this is not the way you do the science. It is not recommended on the standard textbook behavior of science. But sometimes if you took this, if they took the Rosalind Franklin's standard way of these following things, probably they could not have been able to win this race. So simply they jump onto the conclusion, and based upon their presumption, they built the model, and then it turned out that their model looked so beautiful, perfect. So that was all it mattered. So that's another irony to that. So anyway, the summary of their model is several important things can be learned from their model. Is that one thing is sugar phosphate backbones are the outside and the nitrogenous bases are inside of the helix. What do you mean by that? So we can explain such thing in using this portion of the DNA double helix held together through this complementary base pairing between TNA, GNC. So what they believe was this nitrogenous bases which can provide this complementary base pairing were buried inside of the helix, and the sugar phosphate backbone is exposed. Why can you call this as a sugar phosphate backbone? They are all identical, and these are sugar, phosphate, phosphate. These are all identical skeletal backbone of one DNA strand. So this is one DNA strand, and this is another DNA strand, this double helix, okay? It doesn't show as a helix because you are only seeing only very small part of it, but if you look more wider overview, then it formed automatically, spontaneously from this twisted helical structure. So in this cartoonic version of DNA double helix, this letter stair-like bar represent what? Nitrogenous bases and the hydrogen bond, the complementary base pairs, right? And this ribbon, twisted ribbon-like structure represent what? The sugar phosphate backbone, okay? So their assumption and their prediction, and accordingly the built model, according to this model, the nitrogenous bases are inside and sugar phosphate backbones are outside, actually bears a very strong significance in that. At the time, some other more renowned scientists like Linus Pauling, maybe, I'm not really sure how many of you can recognize his name, but Linus Pauling was a very renowned biochemist at the time, and he had already won a Nobel Prize already, revealing after discovering some structure, protein structure, certain particular protein structure. So he was the king of this structure, and then he publicly he always said that DNA is triple helix, okay? And in that structure, the phosphates, these phosphate groups are inside of the helix, and these nitrogen bases are outside, stuck out. That's his view, the Linus Pauling's view of DNA triple helix. But Watson and Crick, when they thought about, if that was the case, chemically that's impossible. Why? Because if you put this phosphate inside together, this phosphate chemically, they are all negatively, highly negatively charged. Remember, DNA, because of this presence of the phosphate group, DNA is highly negatively charged. And we take advantage of this particular property, being negatively charged, in many different molecular technique occasions. Then they reasonably thought, if you put this phosphate group together inside, then they will repel each other, this negative charge. So it could generate highly, extremely unstable structure. So it can be true, even though that was said by that renowned king, Linus Pauling, but whatever, I'm not going to follow. So their idea was, okay, this should go outside so that they should not interfere each other. Rather, these guys can be easily associated with through hydrogen bonds, so they can be easily put inside. That's more reasonable. That's how they follow their logic. Science is the art of logic, and it also shined at that. And The other thing is, they kind of already guessed that this has to be the case, complementary base pairing between these two, or is a purine versus pyrimidine. If that was the case, the dimension. This diagram explains this. So, for example, if you place two purines each other, or two pyrimidines each other in generating complementary base pairing, this dimension will have wavy-like structure. But if you always have this particular, always having one purine matching with one pyrimidine, this dimension always stays the same, so preserving this helical internal dimension constant, which is extremely important in maintaining the stability of the DNA structure. Okay, am I going too far? Okay, so I will shut up. So that was anyway his logic. Okay, and more importantly, and this point is the one actually allowed the Nobel Prize winning of their result. So all these chemical details, a beautiful model which is correctly built, good, fine. But as we have seen in the previous result, like even Avery's experiment, Hershey-Chase's experiment were also very beautiful, meticulous done, but nobody, okay, even thought about, oh, they should receive Nobel Prize. I mean, what difference did they make? This significance in, okay, by presenting this model, people were able to understand, oh, DNA has to be the genetic material. Previously we thought that on chromosome protein was kind of genetic material because of their more potential to be able to generate diversity. However, forget about the diversity. Another very more critical matter is the replicability. As a genetic material, not only the material should be able to generate the diversity, but also another more important thing is should be able to easily replicate. Every occasion of cell division, your genetic material also should be replicated. Okay. So when we refer the self-replication, it doesn't mean that without getting any help from any enzyme, DNA itself can be replicated. No, I do not mean by that. Self-replication means without trying to incur any very inconvenient template, if you are going to replicate something from, you need the original template. So every time you are going to try to replicate, you have to generate some kind of template, and then you have to exactly copy this thing. And protein, if you were to use this protein as a genetic material and do this so-called replication, it's going to be very difficult. But when you look at this DNA structure held together through complementary base pairing, this complementary base pairing is the key. It behaves as when you try to, let me ask you something. When you try to make up for ladies, okay, what item do you really, absolutely have it? When you try to make up, without it, you cannot even make it. Usually, can't do that. It's a small item, but what? Yeah, mirror. Without mirror, how can you make yourself up? So you have to be able to see yourself when you try to put something on your face. Mirror. And this complementary base pairing things behaves as if one strand behaves as a mirror to the others. So when you try to duplicate this, each existing strand will become self-template for the newcomer. That's what it means, self-replicating material. So it easily become a template. Oh, see me. You exactly follow me. And you know how to do it. You know already how to follow me because you and I are going to be linked together through what? Complementary base pair. You know that. So easily new nucleotide can be attached. So the only thing it matters is as long as you separate this existing two strand, and in between these two separated strand, new strand will fill up, and that's how easily you can finish the DNA replication. So by looking at the structure, people, they are not stupid, those scientists, so they knew that, oh, if DNA structure were like this, then DNA replication should go this way. It's so easy and so meticulous, so accurate, it can be done without any problem, was what they have realized. So, oh, okay, it's a great job, so we should give them a Nobel Prize. That was the logic. So this is how you, so starting from this existing part of DNA held through complementary base pairing, as you can see over here, when you want to replicate this, all you gotta do is separate this, melt. Today, I didn't have enough time to get organized myself to collect all those your previous questions. So I may have only one time for going over one question, but anyhow, I noticed that in some of your questions dealing with this thing, and heating, when you heat, DNA will also be denatured, destroyed, and how were you able to use those destroyed DNA in transforming principal experiments? Several questions were like that, DNA. But DNA structure were like this. If you apply heat, the only thing you can affect is this complementary, the hydrogen bond. So temporarily, the two double strands got separated into single strand. That's all you can do by heating. And the beauty about this stability of DNA is if you remove the heat, so put this thing, the environment back to normal temperature, like something 25 degrees, even 30 degrees, even 40 degrees, they will reattach automatically. So we call that as a denaturation. But this denaturation, destroying the structure, denaturation in nucleic acids like DNA and RNA are totally different from those denaturation of protein. In the case of protein, if you apply heat, they are all gone. Their fine structures are all jumbled and you cannot restore, practically speaking. Although even in these days, there are some techniques that develop that even you can rescue those messed up protein structure, but still we have a long way to go. So anyway, at this point, we can safely say that, yeah, protein you cannot. Protein denaturation, you cannot rescue. You cannot recover the structure. DNA denaturation, you can easily recover, like by simply retracting the condition that generated this denaturation. DNA will reform automatically. That's the degree of stability of DNA. So anyway, when you separate temporarily for the replication, then the new strand synthesis can go along with all together, and finally culminating in two new strands. Notice that whenever you have this new strand as a result of DNA replication should be something like a what? Hybrid, isn't it? What do I mean by the hybrid? All these two new strands consist of one old strand and one new strand, right? Instead of one having entirely old strand together and one entirely new strand together, but by the mechanism of this DNA synthesis, you cannot avoid this mixed up hybrid structure of DNA. Is another extra gravy that you could learn out of this. So anyway,
교수님, 근데 DNA의 반보존적 독재를 알아낸 세상에서 가장 아름다운 실험이라 불려요.
who said that? I don't agree. Yeah, maybe to them the most beautiful experiment in the world, but to me, I'm nobody, of course, who would value my personal opinion. But anyway, I don't think that was the most beautiful. There was also a clever idea of this. She was referring the experiment. I'm not going to introduce you, but there was the experiment that showed that this mixture during the DNA replication. And that was really nicely done too, of course. And another thing is this. Of course, this is remotely related with the DNA double helical structure, but we also must not forget about this fact that individual nucleotide in one single strand is assembled through this particular chemical bond called, its name is phosphodiester bond. We don't have to remember phosphodiester bond, who cares about? But anyway, this phosphodiester bond is also a type of condensation reaction, just like the chemical bond that you have seen in the case of macromolecular assembly. Condensation reaction, if you still remember, is a reaction by which a water molecule is extracted and a covalent bond is formed. Is condensation reaction, isn't it? But so there are several different names of condensation reaction. But anyway, this phosphodiester bond is also another type of such condensation reaction. Where are you looking at? Okay, thank you. So because of that, so this is for those, particularly those who took the high school biology but still didn't really quite understand why DNA always goes from, what is this, 5' to 3' direction. Probably you can understand at this time around. So this figure is the way the two different DNA nucleotide can be linked through the so-called phosphodiester bond. Remember, there are two, one H and the other OH. So overall, one water molecule is withdrawn, and afterwards you can have this chemical covalent bond is formed. So now these two nucleotides linked. Okay, so this is the way you put one by one, you put the nucleotide into a existing. For example, if this is growing DNA molecule, by putting more DNA molecule over here, you can grow this particular chain. This is how you go over this DNA replication process. But thing is, from where to where? Maybe some of you actually cannot, it's too much because you have never heard of this type of thing before. But I'm telling you, to those who already took a little bit of high school biology, so when we refer this 5' or 3' position of a DNA strand is something, what it really means is that if you look individual nucleotide over here, let's focus this individual nucleotide structure. Do you notice this particular number, 3' and 5' over here? This is something carbon. In organic chemistry, it's a convention that we put specific numbers on each different position of a carbon in organic chemistry. So that numbering started over here, number 1, 2, 3, 4, and 5. Okay. So that's what it means. But why instead of 1, 2, 3, 4, 5, but 1 prime, 2 prime, 3 prime, 4 5 prime? Because there is another carbon, 6 carbons over here, like for example. So in order to distinguish those carbon, so in the lower part of this sugar, we put the numbers instead of the regular number, but use the primed number. So 1 prime, 2 prime, 3 prime, 4 prime, 5 prime. So carbon position. Okay. So anyway, then once you have that, and if you look at between two, in which two different parts that phosphodiester bond formed, then always from this position having this 3 prime carbon having OH group and another nucleotide 5 prime carbon having phosphate group. So this phosphodiester bond always form between OH and phosphate. The OH of 3 prime carbon of existing nucleotide and phosphate of 5 prime carbon of incoming nucleotide is always this phosphodiester bond is formed. So if you expanding this pattern, then hopefully you can understand why now always the DNA chain will grow from 5 prime direction to the 3 prime direction. Look, so let's say this is one DNA strand. The other way, there is a one phosphate group having 5 prime position and one nucleotide having this OH at the 3 prime position. And new nucleotide will be only inserted, remaining another 3 prime hydroxyl group over here. And another nucleotide will be added, having another OH group. So it will grow from this to that direction, which overall you can say is DNA grows from, in DNA replication, DNA strand expand or grows from this 5 prime, the very 5 prime position to the 3 prime position. And after all, if you examine from any existing DNA strand, only the very end, one end, still have this phosphate group, unreacted phosphate group, and the other end, hydroxyl group, still remain. Only one hydroxyl group, still unreacted. One hydroxyl group remained. That should be 3 prime end of this DNA strand, and the other opposite end should be the 5 prime end, because that is the one carbon that holding the phosphate, and this is the carbon that holding this free, so to speak, free OH group. That's how you conventionally designate the DNA strand. So that is the reason why DNA replication always goes through this direction. Even RNA synthesis also goes through this direction, because RNA synthesis, if you imagine, although I didn't explain it yet, but it is a process of adding nucleotide one by one. And the way you add, connecting, is the only other way, there is only one way of linking this nucleotide together. Phosphodiester bond, which should always be formed between one hydroxyl and one phosphate, having this particular orientation, 5 prime to 3 prime. So that is the reason why always it goes to 5 to 3 prime direction. Okay, so And one more thing. One more thing? No, I still have 15 minutes. This is another figure explaining the procedure of DNA synthesis. We know that this is a DNA synthesis rather than RNA synthesis because of the presence of this T. In RNA, the T does not exist. Instead of T, there is a U nucleotide, uracil exists, so we automatically know this should be DNA strand. One may say, hey, RNA exists in single strand. So that's another clue. Not necessarily, because RNA can also exist in double-stranded too. Because RNA, the ability of this as a nucleic acid, the ability of existing as a double-stranded is totally, solely dependent upon what? What do you think now? Out of this today's lecture, what can you say? The ability of any nucleic acid being able to exist as a double strand is solely dependent upon what? Complementary base pairing. RNA, do you think RNA strand cannot be engaged in complementary base pairing? Why not? They have the same nucleotide, nitrogenous base, which can participate in hydrogen bond with respective partner. So RNA can also. DNA and RNA hybrid is also possible. DNA nucleotide of something G can pair with RNA nucleotide with which one? This case, C can be paired too, right? So as long as they can find their respective nitrogenous base partner and form a hydrogen bond, they can do so. So RNA double helix is also possible. Simply, for some reason, they don't like to do it as much as DNA. But if they had to, they can do with no problem. So anyway, I will just bring that up later, of course. Let's get back to this one. In this figure, one single nucleotide is just inserted over in this position, right? Did you notice something special that you didn't expect? Yeah? One single nucleotide is just inserted during the process of this DNA replication right now, but something, is that what you have just expected or something that a little different from what you have expected? In what aspect? Or should I also put up some incentive over here? Like 1 point incentive? Or maybe 2 point incentive, raising up. Auction today. I cannot go up beyond 2 point. That's my marginal line. 2 point. What? So something strange here. Okay, so I will tell you what. Just I will lower down this the reward as a 1 point, but what about this? Still 1 point incentive is up here. This nucleotide has what? How many phosphate does it have? 3. The nucleotide as a member of this nucleic acid has how many phosphate? So it's 1 phosphate. So this has what? Extra 2 phosphate. Why do you use this extra? This is something like very similar to ATP, isn't it? The only difference is this is not ATP, but the sugar is a little bit different, okay, because it's a DNA. But anyway, The ATP is very similar. ATP is in that respect is another type of just a nucleotide to ATP. Our energy, the universal energy ATP is also a nucleotide. But getting back to this point, why? Why trinucleotide? Triphosphate nucleotide instead of monophosphate. Monophosphate nucleotide M and P, but triphosphate nucleotide is used. Is there any reason we can think of? So because we brought this triphosphate nucleotide as a raw material in the synthesis, so we had to dump, during this insertion procedure, we had to dump this extra 2-phosphate and only insert monophosphate version of nucleotide. Why this inconvenience? Were there any monophosphate nucleotide not available in our cell? Is that the reason why? I don't think so. There's got to be plenty of monophosphate nucleotides must be around, but why only this triphosphate nucleotide? Is there any reason? We went over this concept before the midterm exam we have already. We didn't notice. I'm not trying to bring up the new concept over here. We went over this. Simply, you didn't just realize this. What's the benefit? Obviously. Why bother? Triphosphate nucleotide. Is there any advantage? Instead of using this monophosphate nucleotide, that is exactly the form that you need to put in. But I'd rather have this. I'd rather start with this and then I will dump this extra 2-phosphate during the process. That's how we do it. Why? Okay, so I will take that 1 point incentive myself. Okay, so I already have my cup of coffee, so I don't need any extra cup of coffee. But anyway, I will just try to good thing to myself too on the way back home today. Okay, thank you. It's about energy. Why? Is this process is the what? Anabolic or catabolic reaction? DNA synthesis. Anabolic reaction requires what? Energy. So that must be, there is some kind of ATP and it's an energy source must be provided. Where do you see any ATP? No, you don't. This is an individual nucleotide that is going to be put in. But instead of actually inserting exactly the form that you need, if you have extra, during the process you are doing this clipping, this 2 extra phosphate is what? Is it catabolic or anabolic reaction? It's a catabolic reaction. So, if you perform this catabolic reaction, what do you get? Energy. That energy is being funded over here. So you don't have to try to collect any other extra energy, but simply utilize the original nucleotide raw material's energy. Simply breaking this thing up, you get the energy, so that energy is being used this. That's very clever. So you don't have to worry any extra energy thing is the reason why they do that. Okay? So maybe I don't have any time to go over the student question today, so I will just do the double time on Wednesday, sorry. So anyway, so another feature is this, antiparallel. Is this. So on your left, the strand on your left, which up or down, which area is 5' end? On the left strand, the top or bottom, which one is your 5' end of the, not your, is 5' end of the strand. Bottom, yeah. This is the 5' and this is the 3'. On the right strand, which is the 5'? Top or bottom? Top. So they are obviously held upside down. Okay. Why? Otherwise, this configuration of the hydrogen bond between two complementary bases doesn't fit. Actually, this is the area, the point that Watson and Crick had the most trouble when they were building the model. Originally, they assumed this two-strand double helix were held in parallel instead of anti-parallel, and it didn't fit. So they, what's going on, almost gave up. And one crazy day, hey, what the heck? How about putting one strand upside down and was automatically, beautifully all fits around. That was the final conclusion of there. So naturally these DNA double helix are held in this anti-parallel fashion because of their particular structural configuration, configuration between the complementary nitrogenous bases that should form the hydrogen bond. And it also applies to any nucleic acids that are going to be held through the same complementary base pairing. So between RNA and DNA, also between two DNA strands, they will always should be arranged in this opposite way. Remember, that's another. So anti-parallel is the one. And one last thing about this, Watson and Crick discovered, they specifically mentioned this thing too. So in the original Nature paper in 1953 or something, they mentioned all these things specifically, including their self-replicating molecule capability of specifically they, of course, they realized that when they built. And also this thing. This is one particular nitrogenous base called the cytosine. Here, cytosine. Naturally, this amino group can be detached, and then instead of oxygen can be put in, we call it as a cytosine deamination. It naturally occurs without doing anything. Okay. In the cell, it spontaneously happens. It's just a matter of frequencies. And the problem is, cytosine, originally cytosine, now this is what? This is another nucleotide, totally different nucleotide in uracil. Okay? So this type of small chemical conversion, the point is, you don't have to pay any fine details attention. You don't have to. But simply put, this nucleotide can be spontaneously changed into different form, and during the DNA replication, it can go unnoticed. They simply regard this as a different. Originally, there already this particular nucleotide was, and then during the replication, then a wrong, incorrect complementary base will be paired. Now that's the start of the mutation. So the baseline mutation rate will always there no matter what you do because of this thing. And also on top of that, the enzyme carrying out this DNA synthesis, its name is DNA polymerase, will have its own certain basal level of error. Just like when you dictate something, you generally have some typographical mistake as you do. So does this DNA polymerase. So you can anticipate certain level of always some mutation will occur at each round of DNA replication was there. Okay, so anyway, This gentleman was Linus Pauling that I mentioned, that this was the gentleman, the Maurice Wilkins, that the boss of this Rosalind Franklin stole her data and then presented to him. So these three gentlemen were the one who finally received the Nobel Prize. So what about this Rosalind Franklin? Of course, if she were alive back then in 1962, she would have, of course, also got the credit, due credit. But unfortunately, during her experiment involving all those X-ray exposure, she died of cancer before the actual Nobel Prize announcement. That is the only reason why she was not included in this particular Nobel Prize. So, okay, see you Wednesday.